I'm going to stop sharing now and introduce the next panelist, and we'll hold questions until the very end. So the next panelist on our list is Brian Paegel. Brian has really been an innovator in the realm of on-bead DEL chemistry and microfluidics to allow the utility of those compounds that are separated by bead. And so I really am excited to have Brian here to tell us where the field is going in this space. And I will pass it over to you, Brian.
Thanks, Gwen. And thank you very much for the invitation to speak at this event. And thank you so much, Serene and the NCATS team for organizing this event. I'm just going to launch right into things because this is a DEL conference, so no time wasted. A little bit of background as far as commercial interests. I have a significant financial interest in a company called 1859, which is based in La Jolla, and they are commercializing some of the technology that you'll see in the talk today. So diving right in, everyone's been seeing today how DNA-encoded library technology is an affinity selection-driven approach where your target of interest is immobilized or isolated somehow, incubated with a very complex mixture of small molecules, each conjugated to a DNA that encodes the recipe that was used to synthesize that molecule. The unbound species are washed away, and then the bound species are somehow eluded and sequenced to determine what the hit structures are. And when I started my career at one of the NIH molecular screening centers at Scripps, Florida, I was enamored by this as a way to kind of really get around some of the liabilities I saw in a conventional compound collection, which was the ability to design novel matter of any kind of arbitrary property space and also not have this overhead of having to curate a large amount of individual compounds. But what I saw missing was a way for one to use these libraries to do the really elegant and really high definition assays that are available when you have this type of discovery platform. So biochemical activity assays, cellular assays, and so forth. And so as a person with a background in microfluidics, I saw an opportunity to merge DNA encoding, solid phase synthesis, and microfluidics to reconstitute some of the advantages that are present in a high throughput screening facility. And so the back of the envelope calculation that really drove the genesis of this project in my lab was that if you take a microfluidic droplet of activity assay reagent that only is 100 picoliters, and you put into it a synthesis resin particle that has a loading capacity of 100 femtomoles, and you somehow liberate all of that compound from the bead and into its host droplet, you can generate a very concentrated solution, far more concentrated than one would use in a typical screen. And so this set into motion a kind of multi-pronged attack led by a very talented group of early graduate students in my lab, who I'll acknowledge at the end. One was to develop a solid phase DNA encoded library synthesis paradigm. Of course, this was the first iteration back in the Brenner-Lerner kind of thought experiment, was to do everything on beads. But we had the advantage of coming in later and standing on the shoulders of giants and using some of the really nice approaches that were laid out by Praesis, now GSK, using the headpiece DNA molecule, and just basically adapting that to solid phase to do iterative encoding oligonucleotide ligation onto resin that has been bifunctionalized. So let's say amine sites for compound synthesis, and then that headpiece that's immobilized for DNA ligation. And then just these iterated steps of building block coupling, oligonucleotide ligation, until you install a final primer binding site that allows you to do the high throughput sequencing and structure elucidation that everyone uses to understand DEL output. And the one difference here is that we're building these molecules on solid phase. You can see that we use click chemistry to install our headpiece, but we build our compounds on a photolabile linker, and that's shown here. So that allows us to use UV light to sever the structure that's being screened from the DNA that encodes it. And we've already heard why one might want to do that in terms of escaping from the potential interference of DNA and screen the small molecule natively. And so we also developed microfluidic screening infrastructure that allows us to put these DNA-encoded library beads into a flow of activity assay reagent, encapsulate the beads in droplets, which is shown here. And in the subsequent parts of this cartoon, you'll see that red beads are schematized as the active compound, and then all other colors are inactive. The beads flow in through a photocleavage junction. This is where we have an onboard UV waveguide that is delivering 365 nanometer light to droplets in flow that cleaves the compound from the bead and into the droplet. The DNA, again, remains attached to the bead. It's just the small molecule. This passes into this creamer incubator, which close packs the droplets and allows a biochemical activity assay to incubate. And that assay progress is schematized here as an increasing green color as time progresses in this incubator. The oil is removed from the flow temporarily and then merges back with the droplets at the end of the incubator, where the droplets sail single file through the focus of a laser-induced fluorescence detector. And the assay endpoint is read out in high speed, approximately 100 per second. And then we apply a high voltage AC pulse to an onboard electrode to sort any hit droplets over for collection. And I'm going to show you now what that looks like in movie form. This is the circuit that is used. It's a little more complex than the cartoon you just saw, because in the real world, we have to move things around to make everything nice and compact. But I'll walk you through as we go through the movie what the different parts are. These are the library beads entering in flow. This is a separate assay flow, and this is droplet generation. So here are the droplets forming. There's a little splitter that just increases throughput. The droplets go through the photodosing junction. This is the waveguide over here. The droplets travel downstream to the creamer incubator. Creaming is the process by which you remove the continuous phase from an emulsion and close pack the dispersed phase. The close packed droplets are all here. They're very dark. There's many layers of them. You can see that periodically some droplets escape into the side channels. There's really not that many of them. We're not worried about it. And then they unpack here at the end of the incubator and sail down toward the sorting junction. And at the sorting junction, this is the high voltage electrode that is used for doing sorting. In a collaboration with Alex Satz, who I saw on this call at Roche at the time, he's now at WuXi, we initiated a series of kick the tires experiments to determine whether or not this system could deliver on an activity-based DEL screening goal. And so one of their early targets of interest was the enzyme autotaxin, which catalyzes the cleavage of lysophosphatidyl-cholines to generate choline and lysophosphatidic acids, or LPAs, which signal for cellular proliferation. We can transduce autotaxin's activity if we're interested in looking for inhibitors or activators by putting in this fluorogenic substrate, which puts a fluor and a quencher on opposite ends of the phosphoglycerol junction that mimics the same structure in the endogenous substrate. So in the presence of autotaxin, this substrate gets hydrolyzed to generate fluorescent product. And in the presence of an inhibitor like Pfizer compound 8380 here, which is a 3 nanomolar inhibitor of autotaxin, you would expect attenuation of a fluorescent signal. Okay, so a little shout out to the Assay Guidance Manual, because we used them extensively when we first got into the business of drug discovery and assay development. Self-taught everything reading that manual. So thank you to all the contributors for educating me and my group members over the years. One aspect of developing an assay involves determining whether or not it has sufficient statistical power to do your screen. And the way that's done is taking on a microplate, taking a positive control molecule and a negative and arraying it in a variety of patterns, getting the mean and standard deviation of the positive and negative control signals and plugging them into the Z-prime equation from Chang et al. Here in Journal of Biomolecular Screening. And the idea behind the Z-prime is simply that we are looking for an assay quality that's as close to ideal as possible, which is 1. Less than ideal assays have larger standard deviations and smaller distance between the means of the two signals. As the means separate and as the deviations get smaller, the assay goes to ideality. And at 0.5, this is kind of the called threshold for an assay to have sufficient window to go into a high throughput screen where you would scale up your reagents and so forth. So we wanted to use this parameter because this is well accepted by the community of high throughput screeners, but we don't have plates, we have droplets. And droplet data looks like this. It's these little spikes that are happening in flow. Here's a blow up of them. And for each droplet signal, we reduce it to a single value, which is shown here. And we have a variety of ways of doing this. It could be a statistical mode, it could be a area under the curve, and so forth. But regardless of your method, you can gather by an old school analytical chemistry experiment called flow injection analysis what the assay looks like in droplets, either in the presence of the positive control inhibitor, which again is Pfizer compound 8380 here, or without it. And you can see that we can plot a distribution of fluorescence intensities, which are high absent the inhibitor, but low in the presence of the inhibitor. We can fit normal distributions to both of these to get the mean and the standard deviation and calculate a Z prime for the assay. And this can be done for any kind of assay miniaturization effort to droplets. And since I only had 10 minutes, I know I'm going at warp speed, but I want to kind of show some new stuff. This was published in 2019 in ACS CombiSci, if you'd like to go look at what screening data looks like and how it's interpreted. But to walk through it briefly, this is a transient histogram. It's our transient heat map showing what droplet fluorescence intensities look like on the screen, on the chip, as we screen a drug-like library of 67,000 members that we also built with Roche. What you can see is that at the high number of droplet levels, so it's kind of this orange band, these are all of our negative control droplets. There are many that are empty, so our occupancy is only about 0.1 beads per droplet, so 1 bead for every 10 empties. So most droplets have high fluorescence intensity because they are uninhibited. We use the last 1,000 droplets worth of data to get a negative control signal and then calculate a standard deviation threshold here, 4 sigma below that mean, and then collect as hits all of the droplets that are below that threshold as statistically significantly attenuated in fluorescence and therefore likely to house an inhibitor. And so just in side view here to kind of give you a sense for what this looks like if we just kind of bin along one dimension. This is more or less just a one-tailed normal distribution. We're calling some threshold here and then looking at all the hits that are the outliers. So when we look at hits, it's really no different than what we've seen from other DEL groups in industry. Maybe our approach is a little bit simpler in that we're just looking in 2 dimensions here. It's just a 2-cycle library. And we define hits as high value by whether or not we saw that same hit structure on multiple beads. So a 3 here of K-class means that structure was seen on 3 separate beads in the library screen. 4 means 4 different beads, and so forth and so forth. And as we go up in K-class, the false discovery rate drops theoretically by a factor of 10 in other work that we've done. And so just to look at how some of the high value 2-cycle hits drop out, we're again just looking at for monomers that are potentially interesting or suggestive of active against this target, including things like this dichlorophenyl moiety, and also in the carboxylic acid cycle of these thiazole carboxamides, which were expected. Again, the dichlorophenyl seen in a hit. You can look at this paper if you want to see the validation of the molecules from this study, because I'd like to hop on to some unpublished work working on a similar system that's not looking at homogeneous fluorescence in droplets, but rather fluorescence polarization and ligand displacement assay. So the same geometry of system can be used to do binding assays in solution. And this, although we published this initially with, again, SATs on a receptor tyrosine kinase called DDR1, we wanted to pull off a little stunt and look at HIV reverse transcriptase primer template complex inhibitors. Use our DEL to look at this particular target as a potential challenge, something that would be very difficult to do in affinity DEL mode. And so the idea here is that we incubate with our target a fluorescently labeled oligonucleotide substrate. In the presence of the RT, it binds and we get high polarization, but in the presence of a competitor, we would get depolarized emission. And just like before, we can do assay development where we're just looking at the probe by itself, which has a distribution of very low fluorescence polarizations, or the target engaged with the fluorescence polarization probe, which has a relatively high polarization, calculate Z prime from this to move forward with our assay and screen in droplets. It's basically the same thing you saw previously, but now we have a fluorescence polarization on the y-axis. And here's all of our negative control droplets that have the target bound to the probe and our 6 sigma threshold here and all of the hits below it. This is a separate 55K drug-like library. As we kind of moved on, we've maybe followed with Pfizer to try and make libraries that are smaller in terms of size and molecular weight, and more tractable in terms of property space within the confines of what my little lab can procure in terms of building blocks. But here is an output from an approximately 20 equivalent screen of that library with a variety of conserved fragments that are seen in high value hits. You can see here the K classes now are much higher. We figured out how to increase our efficiency of collecting hit beads. Some of these are in validation now. Similarly, in the position 2, you see like some things that maybe look a little bit like nucleobases, things that are seen in antibiotics even. And as I said, we're validating all of these in off-bead assays now, and that's actually looking quite good. So we're pretty excited about this possibility. So to kind of wrap up in this short time frame, you've seen how we've been able to take kind of conventional library, microplate library, and bust it out into DNA-encoded library beads, where each bead has many copies of one compound and a DNA tag that encodes the structure on that compound, or rather the recipe of it, as people have been saying. And I've shown you how we can take kind of a microplate activity-based or kind of just standard biochemical assay and bust it out into a series of droplets that are interrogated single file in a highly integrated microfluidic device. And the one last thing, I just wanted to kind of tantalize people, because I get the question all the time, let's shove cells in droplets. Why don't you just go ahead and do that? And we have done that. It's not really great. It's not really great because, so especially if you're thinking about eukaryotic cells, a lot of eukaryotic cells are adherent. They don't really want to be in a droplet. And on top of that, there's this statistical nightmare of Poisson beads, Poisson cells. And what you end up is with this mixture of cells and beads and droplets. We lose that really nice negative control droplets, which is so critical even for a cellular assay that has higher noise than a biochemical assay. We lose that negative control signal to lock onto. There's a lot of reasons why this is really not the way to go. And I haven't even gone into the kind of difficulties of prima donna cell cultures that don't want to be in droplets and hate detergents and so forth. And so I'm putting this out there as a little tickle that we've been thinking a lot about this, and this has been our quarantine pandemic project. And we'll be talking about the very first phase of this and how we've been able to adapt our libraries now to do activity-based cellular assays using our libraries at Pacifichem this year in December. So please join us in Hawaii. So to wrap up, I want to thank all of the people that are in my lab now. I actually appropriated Wes Cochran, who did the autotaxin work in collaboration with Marie Malone, who's now at Nurix, and she was my library czar for a good long while. I miss her greatly. Amber Hackler just graduated. She's my guru on optics and did all of the fluorescence polarization work. And big props to Andrew McConnell and Alex Price, who did the heavy early lifting on developing the DNA encoded solid phase synthesis protocol and the early microfluidics work. They're both at 1859. And special thanks to Alex Satz, who was a great collaborator and worked with us to design and build the drug-like library, and provided us with some really interesting screening targets and lots of great conversation along the way. Thanks to my funders, and thanks, Gwen, again for the invitation to talk. Sorry, that was super fast.
No, it was great. It's always great. You're very good at communicating very complex things in very direct ways. So I really appreciate that. We're going to follow up with you with questions later.